Microbial fuel cells, electricity production from carbohydrates. Hello everyone. I am Karen and this is the Trends in Bioenergy Technology course. This presentation gives an understanding of microbial fuel cell assembly and operation. The content of the video includes an introduction, the purpose of making the microbial fuel cell, general principle, elementary demonstration of a fuel cell, microbial reduction of the fuel cell, experimental method, the procedure for the discharge of the cell, results, and conclusion. So let's get started. The two manuscripts cover the topic of electricity generation by microorganisms. This is a fascinating idea. Studies have greatly advanced our understanding of microbial electricity generation. Today we will see how a microbial fuel cell is assembled and works to generate electricity. Microbial fuel cells convert chemical energy stored in carbohydrates to electric energy using microorganisms. These fuel cells can be used to solve many problems such as organic waste management problems. If the waste is added to the cell, microbes will feed on it, oxidize the organic substance and turn it into electricity, giving out a clean byproduct, water. Moreover, it is a biological energy source and doesn't release harmful gases into the environment. This way it can be a source of renewable and clean energy. The general principle of the method. Every organism utilizes the energy trapped in an electron-rich substance, carbohydrate. As a result of oxidation of this energy-rich substance, energy is liberated, which is used by the living organisms. The process is more complex and involves many enzyme-catalyzed reactions in a single-celled organism. It involves successive oxidation-reduction changes, and in this respect, resembles an electrochemical process. In normal microbial catabolism, carbohydrates are initially broken in the absence of oxygen. Hence, electrons are stored as intermediates and can be used to fuel the like processes. Ultimately, oxygen acts as an electron sink and takes up all the electrons, completing the reaction. This principle is used for microbial fuel cells. Here, a mediator is added to the solution, which takes the intermediate electrons to the cathode. Since the mediator becomes negatively charged after interaction with the anode, they act as negative charges moving around the cell, hence, producing electricity. The oxidizing, positive, electrode acts as the electron sink. The oxidizing agent used is a simple oxidizing agent, such as potassium ferrocyanide. The current generated is detected by a microammeter. Elementary demonstration of a fuel cell. There is an anode compartment and a cathode compartment in a fuel cell. The former contains a carbon electrode, carbohydrates as the substrate for bacteria, a mediator, usually theanine or methylene blue, and a pH 7.0 buffer solution. The latter contains a carbon electrode, potassium or sodium ferrocyanide, in a pH 7 buffer. Both compartments are separated by an ion exchange membrane. Different types of microbes can be used to construct the cell, keeping in mind that the microbe can produce energy efficiently and is not hazardous. For example, E. coli is a very suitable organism but is pathogenic. Yeast may be used but is not very active and is sluggish. A preliminary experiment. Microbial reduction of the mediator. First, it is necessary to check whether microbes are active or not. Suitable redox mediators are theanine or methylene blue. They are water-soluble and can easily be absorbed onto the surface of the electrode. To check the microbe activity, a solution of microbes suspended in a buffer of pH 7 is taken in a test tube and a small amount of mediator is added. The color will turn blue. However, as the reaction starts, the solution becomes colorless because of the reduction of the mediator. If the blue colors maintain, it means that microbes are not reducing the mediator. Moreover, if the oxygen gas is let into the cell, it makes the solution blue again. It is because of the oxidation of the mediator. In the absence of a mediator, cells work for some time, because of the residual current, but the voltage falls rapidly. The mediator is added carefully by a syringe needle. As a result of adding the mediator, the current rapidly increases and the blue color of the solution fades. When the voltage reaches its maximum, now the cell is ready to conduct electricity. Electricity production from carbohydrates. This paper investigates microbial fuel containing Proteus vulgaris. Also, the addition of glucose using a computer control system and single addition of the glucose is investigated in this paper. Experimental method. The anode material used is graphite felt. This electrode material is chosen because of its low resistance and its advantageous physical structure, which consists of an open network of fibers allowing free access of organisms and mediators to the electrode surface and into the solution. For short-term experiments, the cathode is of reticulated vitreous carbon, used in conjunction with potassium ferrocyanide solution. When current is produced over an extended period, the ferrocyanide concentration falls, leading to a change in the cathode potential as a result of the change in redox ratio. Consequently, in long-term experiments, an air cathode of platinized carbon paper was used in a concentrated buffer to provide a more stable potential provided by the well-known oxygen electrode reaction in which oxygen is reduced to OH negative. A platinum wire was attached to the electrodes with a conducting epoxy resin. Mediator. A perfect mediator would be stable in both the oxidized and reduced forms, and would not decompose during long-term redox cycling. In order to increase the coulombic efficiency of the cell, the mediator should accept electrons from as far from the electron transport chain as possible. The mediator preferred for fuel cells that run longer than 24 hours was 2-hydroxy,1,4-naphthaquinone, HNQ, which is soluble, stable, and much less prone to adsorption. A platinum wire is attached to both the electrodes to connect them. Preparation of bacteria. Proteus vulgaris was the preferred biocatalyst. They were cultured aerobically in nutrient broth with no extra glucose at 37 degrees Celsius for 18 hours on a rotary shaker at 200 rpm, washed, and resuspended in a potassium phosphate buffer. Preparation of immobilized bacterial. Electrodes. Bacteria were immobilized onto the electrode surface by the formation of an amide link between oxidized carbon atoms on the electrode surface and amino groups in the bacterial cell wall using the carbodiamide technique. Felt electrodes were activated by soaking in 1-cyclohexyl 3-carbodiamide methylbutylene sulfonate solution with constant agitation at 4 degrees Celsius for 5 hours. They were rinsed in water and potassium phosphate buffer with pH 7 and were then exposed to a suspension of Proteus vulgaris with constant agitation at 4 degrees Celsius for 12 hours. Excess Proteus vulgaris was then washed off with cold buffer, and the electrodes were used immediately. Fuel cell assembly. Experiments were carried out in stacks of cells built up in rectangular designs. Each cell unit had an anode and cathode separated by a divider. Each compartment has inlets for the addition of the solution. The stack temperature was maintained at 30 degrees Celsius. Procedure for the discharge of cell. Initially, the anode compartment contains only potassium phosphate buffer. The cathode is set up in potassium ferrocyanide. Voltage is measured using a voltmeter. When voltage is stabilized, the mediator is added. When the voltage reaches its plateau value, the cells are connected to their load resistance and the current was measured. To test the current-producing capabilities of the cells, they were allowed to discharge at different known resistances. The voltage is allowed to recover to a plateau value. The voltage and the currents are measured instantaneously. Results. Out of the 4 cells, in anodes of the 2 cells, bacteria were immobilized and in the other 2 dry mass of free bacteria was present. One cell of each type was fed with glucose and the other was not. As the mediator ratio increases, current increases, and when it lowers, voltage also lowers, stabilizing the current at the plateau value. The current produced by the free bacterial cell rose more rapidly than the immobilized one, reaching the peak value earlier. However, the plateau for the immobilized cell was 7% higher than that of the free case. This may be due to the location of the bacteria relative to the electrode surface in the two different configurations. The reduction of mediator by immobilized bacteria can occur only at the electrode surface, whereas in the case of freely suspended organisms, it may occur throughout the anode electrolyte. On the addition of glucose to bioanodes containing immobilized bacteria, the mediator must travel to the electrode surface before it can be reduced. Computer control performance of free and immobilized bacteria. In the case of the computer control system, a threshold voltage is set. Like in this case, the voltage was set at 0.4 volts, a current of 0.4 microammeters, and the load resistance was 1000 ohm. When the voltage drops below the threshold value, computer recognizes that and more glucose. As a result, current increases. This way of adding the feed to the system is more efficient than adding all the glucose at once. Because access to glucose can cause incomplete oxidation of the substrate. Moreover, when pH drops, the computer automatically compensates for that by making the anode more positive. In this experiment, pH was measured every 24 hours. When pH drops below 6, the buffer was replaced to protect bacteria from over-acidification. Conclusion: immobilization of the bacteria improves the movement of mass transport of mediator between electrodes and a solution. Hence, produce a constant current. It is seen that immobilization also increases the lifetime of microorganisms. It is noted that pH stability and the mediator can be improved in the future. These aspects are currently under investigation. With present design, currents attainable are limited to 1 ampere, may be due to design factors and the electrode and internal resistances. Thank you so much for listening. I hope this presentation will be very helpful in understanding the workings of a microbial fuel cell. Have a good day.